Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Bioanalytical Support Biosimilars. Expect the Unexpected. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion, a global leader in early clinical research services, offers a unique combination of medical expertise, clinical operation experience, and scientific excellence that gives their clients the confidence to make fast, accurate decisions about their drug development path. For over 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety, tolerability, pharmacokinetic, and pharmacodynamic studies in highly controlled clinic environments. Their founding mission is to help their clients get their drugs to market quickly so that they touch the lives of our family, friends, and people in need around the world. And now it's my pleasure to introduce you to your speaker for today's webinar, and she is Dr. Petra Struve. She's the Executive Director of Bioanalytical Services at Celerion facility in Switzerland, a contract research organization supporting biopharmaceutical industry drug development. She holds a PhD in chemistry and is an experienced scientific leader with more than 20 years working in the bioanalytical and biomarker segment of the pharmaceutical industry. In her current role, she is in charge of both small and large molecule method development validation and sample processing in an extremely automated and compliant environment. Now it's my pleasure to pass over the controls and mic to our speaker, Petra. So Petra, when you're ready, you may begin.
Thank you, Sonia, and hello and a warm welcome to everyone. I really appreciate your interest and thank you for your attendance today at this webinar. In the upcoming minutes, I want to share with you some of our biosimilar experience with the focus on the associated bioanalytical methods. Why are biosimilars still of global interest? 6 years ago, we presented our first biosimilar webinar related, and as you can see, we supported quite some projects in the previous year. Within these, we experienced some challenges, which I want to share with you in the upcoming minutes. And I apologize upfront that I will not refer in the real-life examples to the associated drug names to ensure confidentiality, but I can confirm that the majority of these challenges are independent of the individual drug themselves. But going back to the question, why are biosimilars still of interest? When you look at the top-selling pharma drugs in 2019, you can see that the majority of these are biologics, mainly monoclonal antibodies. The ones presented here are the originator sales numbers, but several of these are already out of patent or exclusively, and the associated biosimilars are available on the market. In the previous 2 years, this biosimilar market climbed up, and for example, more than 40% of last year's bevacizumab, which is Avastin, or trastuzumab, which is Herceptin, prescription included the biosimilar instead of the originator. Europe, with around 60 biosimilar approvals, in parallel with the US with almost 30, is from the history a little ahead of the game, which the following graph nicely presents. The regulatory pathway for biosimilars was first established in the European Union in 2004, which enabled the development and approval of biosimilar medicine based on comparative, analytical, non-clinical, and clinical data. that confirmed them to be equally safe and effective as their reference products. And as a result of this, the first biosimilar medical product, Omnitrope, which is somatropin, was approved by the European Commission in 2006. The US didn't implement any regulatory framework for biosimilar evaluation prior to the enactment of the Biological Price Competition and Innovation Act in 2009. Sandoz filgrastim biosimilar Xaxio received the first US approval in 2015, and at this time already 9 filgrastim biosimilars had been approved in Europe, dating back to multiple authorizations since 2008. Looking at today's prescriptions, more than 70% of these administered one of these biosimilars instead of the originator Neupogen. But what is a biosimilar? Todd Lester from Bioanalytical used in one of his talks a really nice example I want to share with you. He expressed within this the difference between a small molecule and a generic versus a biologic and a biosimilar with quite easy pictures. You can imagine the small molecule via a generic he compared with 2 1x2 beams, which are the same, otherwise your construction, for example, from your house might fall in pieces. A biologic and a biosimilar is like having 2 trees in the nature. They are of similar type, but never equal. Okay, I emphasize in nature because I agree that it does not apply to a perfect trimmed box tree if you have a highly qualified gardener, but this is a different story. So back to biologics. Beside the complexity and the size of a biologic, take for example a monoclonal antibody with a size of around 150 kilodalton. The manufacturing in a living system is expected to end with variations of the associated end products. The outcome will always be similar, but not identical or equal. Therefore, challenges in biosimilar development are quite different from reproducing small molecules via a chemical process, and also the regulator considered this for the biosimilars. They expect that new biosimilars has demonstrated high similarity in biophysical properties, in pharmacokinetic and pharmacodynamic, in safety and effectiveness to an approved biological reference drug. How should we approach this? Because biosimilars are not generic drugs. They have a structure 200 to 1,000-fold larger than a generic, as you can see in this picture I presented here. And they are produced, as mentioned before, in a living system as a cell or a virus. So the development path of a generic is since several decades standardized. We all know this, and requires, besides some analytical similarity testing in the CMC, one bioequivalence trial to be ready for submission. But due to the complexity of the biologic, it is not surprising that more testing is required. And I show this now on the next slide. The authority, they defined the process as totality of evidence. And the authorities really expect that the biosimilar is compared with the originator-related structure and function, animal toxicity, human pharmacokinetic and pharmacodynamics, clinical safety and effectiveness. In at least one clinical trial, a comparison of the immunogenicity of both compounds should be included. And in all of the highlighted ones at the left, the bioanalytical assays can have a significant impact on the outcome of the assessment supporting the submission. But as you can see on the pyramid, it all starts with a basic. It all starts with a CMC testing, which is a foundation for all further tests, and because a lack of analytical similarity can't be compensated with anything built on this. So as a bioanalyst, what we usually see is a final release test of the provided drug, meaning the certificate of analysis. But if you look at the total CMC testing, that's in reality the tip of the iceberg. The complete CMC testing includes an extended characterization to demonstrate that the biosimilar is highly similar to the originator. And within these tests, the biological product is characterized using state-of-the-art technology with a focus to detect any differences between both compounds. The tests focus not only on the physical chemical properties, but also on the functional ones. These are assessed considering the known mechanism of action. Product-related impurities and modifications are identified, quantified, and compared using multiple lots of proposed biosimilar for an assessment with the originator to see if there's a potential impact on safety, purity, and potency. But please consider that any impurity in the biosimilar, which differs from the originator either in amount or structure, should be included in the setup of the immunogenicity testing, but later I will go more into detail related to this. The final released biosimilar drug substance is a starting point for the associated bioanalytical development. The key question is here how to set up assays generating comparable results, considering that minor differences related conformation or glycosylation can have a direct impact on these assays, for example, on the binding capability of the included antibodies. So what are our main challenges for these assays? On a high level, when detecting concentrations of biosimilar and originator, the four main contributors for difference are subject-to-subject variability, lot-to-lot variability in drug concentrations, chemical differences affecting the assay, or precision of the ligand binding assay. The first two are outside of the bioanalytical control and are mostly taken care of by either the study design or a thorough characterization of the material used in the trial as described before. The next two are in the control of the bioanalyst by choosing the proper assay format, which is usually a ligand binding assay, and pushing the assay to the precision limits with support of automation. The big question for all of us is always, what is the proper assay format? In the beginning times, the two-assay approach was the approach of choice, especially when the assay for the originator was already established. With this approach, a separate developed method for the biosimilar was validated and cross-comparison of quality controls were added to both assays to show a bioanalytical similarity. The challenge of this format is that even with this comparison, it remains two assays with different properties. And due to this, the between-assay variability could heavily impact the reliability of the head-to-head comparison, which is the outcome of the clinical trial. And additionally, you have to analyze all samples in blinded studies twice with both assays, which has an impact on the timeline as well as on the costs. With the years, the one-assay approach became the industry standard, as this was published in 2014 by the Associated APS Working Group with the lead author Similarity. A single assay which can quantify both components using the biosimilar and for the applied calibration curve and quality controls. Only one assay is to validate. All samples of a blinded study are analyzed once, and because it is a single assay, the assay variability is reduced, supporting the comparison of the biosimilar and the originator concentration. But to end with such an assay, a thorough assay development is mandatory to ensure that the minimal differences between the biosimilar and the originator are appropriately addressed. For this method development, we go in a two-step approach. First, the bioanalytical similarity, which is not the analytical similarity we discussed in the CMC part, is demonstrated between the standard curves of the originator and the biosimilar. Key reagents might differ between both compounds, and therefore assay conditions have to be optimized. To get parallel or ideally superimposing calibration curves, the focus in the development phase is on the assay methodology, the included critical reagents, the assay design, and the used calibrators. Please note that regulators don't expect that the same assay is used as for the original submission. State-of-the-art technology should be applied. But when choosing the appropriate assay platform, the inherent nature of and the properties of the molecule should be considered. And in parallel, you always have to keep in back of the mind the assay robustness covering the complete development process. At the end of the development, preferably we have parallel or ideally superimposing curves as presented here. A critical comparison of these curves is needed. There are currently several mathematical methods proposed, but I'm a visual person. Already at a visual inspection gives you already a good idea if a one assay approach which will work as this did for these curves I present here. But another assay or another result can be that you have perfectly parallel curves, which gives us a hint that there are concentration differences between both compounds. We can work with that because we can compensate this via a factor. But you also can have consistently non-parallel curves. Here it is suggested to conduct an investigation, as I will show you on the next slide, or to use two separate assays. But for these you need to have appropriate justification. In this first example, real-life example, we have non-matching curves for the originator and different lots of the biosimilar. After multiple tests at our end using different reagents, material, analyst, pulling her hairs out of our head, the only remaining root cause could be an interference not visible in any of the release tests. There were physical, chemical, and biological characterization tests presented on the associated certificates of analysis. But when we looked at them in a second time, we saw that they were prepared for the originator and the biosimilar by different labs. So we really struggled with this, and at the end then we determined the total protein concentrations by using the BCA protein assay of the individual drug substances. And then we saw that all the biosimilars had a different protein content comparable to the originator. When we applied the correction factor, as you can see at the bottom part of the slide, all the curves were within less than 5% and therefore overlapping, and we could run with a single assay. But not only this type of concentration or interference could have an impact. Another reason I want to show you in our second case. For this other program, there was a discussion with a bioanalytical assay, and when we started this, there was no drug substance left because everything was already changed to drug product. And so the sponsor provided us with drug product for the biosimilar as well as for the originator. According to the certificate of analysis, we had received comparable concentrations, but the originator was packed in glass vials, in difference to the biosimilar, which was packed in plastic bags ready for infusion. Comparing both drugs didn't result in any parallel curve, independent of the assay we tried, who did it, how we tried it, how often we repeated it. It didn't work. We invested really quite some time, but it always ended up in the same as you see on the top end of the graphs. And we went back to the sponsor, and the sponsor gave us from their new lot of the biosimilar drug product, the biosimilar compound, and when we implemented this in our process and repeated it, we got parallel curves. And with these overlapping curves, we supported the single set assay, but also because we had this early discussion with the sponsor, it was figured out that we couldn't exclude absorption to the plastic bag. And therefore, the packing was adapted before start of the clinical trial, and this was performed successfully. So proper certified drug substance is a key for a successful bioanalytical PK method, and an early start of bioanalytical method setup might give additional add-on information which can support successful clinical trials. So when we have a perfect developed method, the next step is the validation. And in the validation then, we provide the formal proof that the method is capable of equally quantifying both compounds by confirmation of the comparable reactivity, a systematic comparison of the quality control samples, and a direct comparison related parameters as selectivity, sensitivity, dilution, linearity, and stability. But the key experiment of each biosimilar PK method validation is a test related to the limit of similarity. Within this test, multiple dilution by multiple operators on several occasions are performed using 5 levels of quality control samples and applying industry acceptance standards for accuracy and precision. These are 20% for the low, mid, and high QC, and within 25% for both ends of the calibration curve. Biosimilar and innovator are analyzed in a tandem, as you can see on the presented plate layout, whereas the green one is the originator and the violet purple one is the biosimilar. And this layout is chosen to uncover trends visible in difference with the biases between the results we receive. And even that the industry standard allows today 20%, regulators today expect, as we learned a few weeks ago when we prepared for a new biosimilar program, they expect a bias of 10% between the both compounds. And you think about it, oh my God, that's half of what was accepted before. But we went back to our old study data. And you can see there that the 6 plates of the same plate design are processed. So you're ending up in 18 individual values per concentration level for the both compounds. An inter-batch comparison between the data of the reference and the biosimilar is used for the comparison. And for each concentration of the biosimilar, you compare this with the respective concentration of the same level originator, and the individual as well as the overall mean bias is calculated, which should be within the original 20% and within the 25% for the ends of the curve. Also, the inter-batch bias should be within less than 30% for the high, mid, and low QC, and 40% of the upper and lower end of the curve. But as you can see, and I now refer back to the boxes, the colored boxes I put on the top end, the mean bias for our innovator, which is the one in blue, is easy within my 10% or 10 to 11%. The CV also. For the biosimilar, we have comparable results, but the important box is the green one. The green one gives you the comparable biosimilarity results, and they are maximum at 6%. So even years ago, when we implemented the biosimilar assay and the biosimilar support, we already were in this time what the regulators today expect. But still, at the edges of the curve, there is always a higher imprecision than in the middle of the curve. And to ensure that the bioanalytical impact on the head-to-head comparison is as minimal as possible, we pre-dilute samples, ending then in concentration in the middle part of the range. And that this is a successful strategy, I show you in the next real case. In this program, our sponsor really needed a high statistical power to compare both drugs. Due to this, a 2-stage clinical study design was planned with 170 and 100 subjects respectively. From a bioanalytical end, we try to treat the samples from the originator and the biosimilar equal, meaning we analyze the samples for the 2 periods per subject in one analytical run to minimize assay variability, and parallel with a proper dilution of the scheme to determine all the sample concentration in the middle of the curve. This approach ended in a faster and cheaper submission because the statistical endpoints were met already at the end of stage 1, resulting that the second stage was for go and the submission happened 5 months earlier. So now I end with my PK part because another important part of each development is the immunogenicity. And when we talk about immunogenicity, this occurs when the immune system recognizes the administered test item as a foreign and induces a humoral or cellular immune response. The immunogenicity of profile of a biologic can significantly alter by even small differences in its manufacturing process that are accompanied by changes in the product attributes, like structural properties, glycosylation, aggregation, impurities, or contaminants. But also changes in the dosing schedule, the administration route, or the patient population might impact the immunogenicity profile of a drug. And this can be a significant concern as it can affect both safety and efficacy, meaning the impact can range from apparently no effect to changes in the pharmacokinetics, loss of effect, or serious adverse effects. Assessment of immunogenicity is therefore of critical importance when establishing safety profiles for biologics, and head-to-head assessment of immunogenicity between the biosimilar and the originator should be considered as part of the clinical development program. So, and even that the current guidelines give a prospect of the expected outcome, none of them really clearly describes how we should do this and how we should set up the assays that both drugs can be analyzed in the same way. So the goal is to evaluate potential differences between the biosimilar and the originator and to provide a meaningful interpretation of comparability study data, and not only to ensure safety and efficacy, but also to allow for substitution and exchangeability. The agency's recommendations are that the biosimilar is equal or less immunogenic than the originator. The challenge is that ADA assays are inherent qualitatively in nature, with 4 main assessment stages presented at the right side, which include screening. Positive samples are then afterwards confirmed. Then we go via the specificity to the characterization and do something like a titer determination, and also relate the neutralizing effect. The response is usually yes, no, without a standardization, and this is without a standardization because there is a general lack of available international reference sera or standards which we could use. And this is a difference to the previous discussed PK assay. Therefore, immunogenicity data are highly dependent on the assay we use to measure the ADAs. While there is no single assay format suitable for assessing the immunogenicity of all biologic, regardless of the method selected, all assays must be thoroughly developed and validated. Last year, the AAPS Biosimilar Working Group has published a white paper to guide the industry on best practice and testing strategy when developing immunogenicity assays for biosimilar programs. And as mentioned before, the objective for the immunogenicity assessment is to demonstrate similar ADA rates for the biosimilar and the originator in the same trial via a head-to-head comparison using the same assay. And for this assay, the regulator expects that the most discriminating assay will be utilized using state-of-the-art technology. No one of us should be concerned that the increase in sensitivity and specificity using the next generation of platforms or methodology ends in an increasing rate of positive samples in comparison to the originator submission. That's why you do the head-to-head comparison. And early on in the development, positive controls against the biosimilar and the originator should be generated in the same way. This is recommended considering that, for example, the FDA and the EMA expect that the biosimilar is not more immunogenic than its originator. But please also keep in the back of your mind that if the biosimilar has significantly lower immunogenicity than the originator in the clinical trial, you have to compare several positive controls in the assay to justify these clinical results. And optimizing the assay by using the biosimilar positive control in a one assay setup will also be the most conservative approach because it will ensure that all ADAs against the biosimilar are reliably detected and that all samples can be analyzed in the one assay format. The disadvantage is that ADAs against the unique structure of the originator might not be detected, but this can be overcome by usage of the positive control generated for the originator in parallel for the assay setup to demonstrate that the positive controls bind equivalently to the biosimilar and the originator. So how to set this assay up with a focus to demonstrate biosimilarity? At the beginning, we perform 2 key experiments, which I will show in the following. As a first step, a side-by-side comparison of the labeled biosimilar and the originator reagents with the available positive controls will be performed. For this, the biosimilar and the originator are labeled side by side, and you have to ensure that the label incorporation ratios are similar for both. If not, relabel if required, because small differences can impact the assay immune response detection ability. If you have this done, as you can see, it can work out into parallel curve. But in this assay, the Assay response of the anti-biosimilar positive control was clearly higher in both assays than the originator, and this difference was caused by the variability of the individual rabbits which were immunized with the antigen. Both positive controls, originator as well as biosimilar, were produced at the same time in the same animal facility, and there still this variability was seen. But the fact that the ratio anti-biosimilar and anti-originator is almost the same in both assays, independent if we use labeled originator or labeled biosimilar, the single assay approach was still feasible, because the differences in the response of the two assays are considered to be within the expected range of the different lots of labeled antigens and are in detail assessed in the associated method validation. The second key experiment in regards to proof of biosimilarity is an extended free drug tolerance test, ending in associated drug competition curves demonstrated that the antigen is equivalent. It is expected that samples containing biosimilar originator will exhibit interferences due to competition between the drug in the sample and the drug used as detection reagent in the assay system, hereby producing false negative results. In the assessment, different levels covering the whole range from the free originator and biosimilars are tested on at least 3 different levels of positive controls. Typically, overlapping quenching curves are regarded as key proof of biosimilarity, as you can see in these graphs. Therefore, a single assay is possible, and these are really beautiful graphs. But there's always a risk of missing putative positive antibodies to either drug depending on the assay format. But as mentioned before, our recommendation is to use the biosimilar positive control in combination with the biosimilar for collection and detection to ensure that all epitopes of the biosimilar are picked up, which might be slightly different compared to the originator. Often we get the request, in the interest of time and cost, to start with a commercial positive control. But with this example, I want to show you that the production of the appropriate anti-biosimilar positive control has another benefit. These 3 to 4 months are really well spent, because the sensitivity for an ADA assay is defined as the lowest concentration at which a positive control antibody preparation in undiluted matrix is consistently providing a positive signal in the assay. In the assay setup for this, the conjugate ratios are optimized to achieve the lowest sensitivity. Our experience demonstrates that often with the anti-biosimilar positive control, a 3 to 10-fold lower sensitivity could be reached. As you can see this clearly on these graphs when you compare the left side with the right side. With the commercial positive controls for the originator, independent if mono or polyclonal, we often struggle to reach the 100 nanogram per milliliter sensitivity as given by the regulators. For our other drugs, we go down with the biosimilar positive control up to a factor of 10 lower. One other lessons we really learned from the programs we supported until today is that when you set up the assay, please ensure to include in the confirmatory step each individual moiety. This can be for a pegylated component to include the PEG molecule or any interference generated during manufacturing. These can be ones which are not known from the originator or which are observed in the originator but at much lower concentrations. Or they can be a slightly different glycosylation comparable to the originator in the biosimilar. So do not only do the confirmation versus originator and biosimilar, include as many moiety as possible into the confirmatory step. And I bring this up because we experienced several times that additional tests were required, mainly related immunogenicity, after the complete package was submitted to the authorities. During the review of the CMC package, differences to the originator were flagged by the regulators, and including these early on in the ADA assay setup can avoid fast corrective actions, or even worse, repeat analysis of study samples required to address potential 80 or 20-day questions. But we also experienced that it is recommended to include in the assay setup preferably the drug substance lot, which is planned to be used in the clinic. Bringing in the bioanalytical assay setup in an early stage can support the process because minor compound changes which are not picked up in the release testing of the CMC could impact the bioanalytical assay. And also this can influence the binding capability of the drug, as you can see then on the next slide. Because this ADA assay setup we applied successfully for 2 different biosimilar developments. Both of them were approved by the regulators in a single go. We were certain we know what we are doing. Beautiful overlapping curves. But there's always this one assay which brings you back to reality. Independent of the positive control, as you can see on the right side, we did not manage to get parallel curves when we included the drug moiety. We assumed, because you can see on the top level, you have the 2 times the biosimilar, and the bottom, the violet and the red ones, this is the originator. We couldn't get it overlapping, but we could get different lots of the biosimilar overlapping. So we assumed that this variation is in one of the moiety of the whole molecule, which wasn't picked up in the CMC release testing, but we never got a final confirmation. It's one of the few cases where we had to choose a 2-assay approach because we couldn't fit it into a single assay approach. But this hopefully highlights what I tried to express before, that considering both the analytical and the bioanalytical similarity can support a faster, successful biosimilar development. And this statement is also supported by an industry paper which was published in April last year. Martin Schiestel and his industry colleagues reviewed and compared the public available information of all biosimilar programs submitted to FDA and EMA until November 2019. Within this paper, they questioned the need of the comparative efficacy study, which was in the totality of evidence pyramid on the top level. But I want to focus on the project that failed biosimilarity in the first approach. They looked in total at 42 biosimilar development programs. And 36 of these met their PK equivalent margin and the immediate go. 6 of the 42 initially failed. The probable reason for not showing bioequivalence included problems in either the clinical study design or an underestimated variability of the serum concentrations. But all of them obtained approval after repetition. If you go to the efficacy studies, 36 out of the 38, the comparative efficacy studies did not really add value to the scientific review process related to the PK part. But 2 out of the 38 failed to demonstrate comparable immunogenicity, and subsequently required further optimization of the manufacturing process to improve product quality prior to obtaining approval. And 3 of the 38 failed to meet their primary endpoints, but obtained approval either with post-hoc analysis or additional scientific justification to close the open question and gaps. With these examples, I want to show that on the one hand, the importance of the bioanalytical assay for the outcome of the biosimilar development, and that we as a bioanalyst should also have this in the back of our mind for any assay development. The better assays we provide, the more certain the primary and secondary endpoints can be met at an earlier stage of the development. But on the industry level, it would be wise to consider that the second pair of eyes using the biosimilarity additionally to the analytical similarity can improve the development. Because within this setup, minor changes happening during scaling up the manufacturing process and not caught by the release testing might raise an attention at an earlier time in the development process. With this, I come to the outgoing view saying, biosimilar development, what's next? What's coming up next for biosimilars? We already spent a few years on these. There are several biologic, including in the graphs at the beginning from today's session, which will become either out of patent or exclusively in the upcoming years up to 2028. And in the near future, this is pertuzumab, denosumab, ranibizumab, or ustekinumab. But another group of compounds are antidrug conjugates, which have emerged as a highly promising new class of biopharmaceuticals. The first approved one will soon lose their patent exclusivity protection, and these will bring biosimilars to a new level. I mention this because an ADC is composed of an antibody conjugate to a cytotoxin compound via a chemical linker. So for the originator submission we are working on, we work in general with 3 types of bioanalytical PK assays, which reflect the total antibody, the conjugate, and the unconjugated drug, as you can see here on these graphs. And now imagine we go to the next level, because the challenge will be how to set up these assays up in a biosimilar setting, considering the different moieties which are involved, and how to approach them in a biosimilar approach. I just try to express this here on these graphs by taking and imagine now, so that gives us, at least for each of the 3 assays, the evaluation of the biosimilarity. That's a new challenge, but also not only the PK assay will be interesting, also the ADA assay requires some thorough evaluation before we start with these, because that is a typical ADA approach for the originator submission of an ADC. And now we have, beside the 3 parts of the ADC for the characterization, we have now the biosimilar additionally. Therefore, this will be the next level of challenges. I'm really looking forward to share our experience related this with you at a later time point. And with this, I will come to the end. And I hope that with the presentation I showed you that to support the reliability of data in a head-to-head comparison, apply whenever possible the one assay approach using the biosimilar. That the importance of proper certification of the biosimilar and the originator is really key to all our assays. That the positive controls generated from the biosimilar can ensure that all epitopes of the biosimilar drugs are picked up during the assay setup, and as we learned more than once, support better sensitivity and immunogenicity assay to ensure that all moiety of the biosimilars are included. And that the bioanalytical assay setup for sample analysis can have a direct impact on the statistical endpoint of the trial. And with this, I thank you for your attention and appreciate any questions if we will have some time.
Well, thank you very much, Petra, for that very insightful presentation. I hope everyone enjoyed it. And with that, we are going to put on our webcams and invite the audience to send in their questions right now. Hi, Petra, to send in their questions right now. We've already received some questions already from our audience members, so I'm going to start with those. But once again, audience members, please go ahead and send your questions through that questions window. So Petra, my webcam is taking a little time to get on, but we'll start as we just need you. Okay, so here's our first question. Okay, here it is. Do you ever experience any issues applying the single assay approach in a submission to the EMA or FDA?
That's a very good question, and a comparable one was asked within the EBF community just a few days ago. I can say based on our experience that this approach was applied in multiple approved biosimilar programs in the US as well as in the EU, also supported out of our company. And this assay setup is really based on the paper of the APS Biosimilar Focus Group, which was published this in 2014, which is this Marini paper, and it's really widely accepted in the industry. The key point of the one assay is really to demonstrate the bioanalytical similarity for the reference product and the biosimilar in the assay. And this also what, if we had any regulatory inspection related this, we, but also colleagues out of the industry experience that really the regulators focus on this biosimilarity, this bioanalytical similarity in their review of the data.
Okay, thank you very much, Petra.
Here's our, hopefully, so otherwise come back if you want to have more insight.
Thank you for that answer. Here's our next question. What are the advantages and disadvantages of two assay versus the one assay approach?
I explained this a little bit in the previous slides, but I think if you have a two-assay approach, and even if this was quite common in the past when we had already the originator validated and the biosimilar was just an add-on, and you did this comparison of the quality control samples using both standard curves, it's still, at the end, it's two assays. So therefore, any assay variability due to the different reagents, due to the setup, has a direct impact on your head-to-head comparison. And if you want to reduce the analytical impact on your, at the end, clinical outcome, the head comparison, it's really strongly recommended to analyze all the samples within a single assay. And don't forget, if you use a single assay, the analysis can be performed blinded. And you don't have to analyze all samples using two assays, which has also an impact on the timelines until when we have final results. And therefore, I think the benefit is really, if possible, use a one assay approach. And if the biosimilar is really analytical already, similar to the originator, we experience that it's possible and that it can work out. Thanks.
Thank you. Here's our next question, Petra. If a biosimilar is already approved in Europe, what additional tests are required for an FDA submission based on your experience?
I'm not the regulatory expert, but I can share with you what we experienced since I got involved in this area. So both regulatory agency, EMA, as well as FDA, they have comparable expectations related to the type of tests to be performed. But please correct me if I missed something there. But the overall biosimilarity is really the performance. And both regulators also expect that the approved reference product, which is approved in their jurisdiction, is used to demonstrate this interchangeability. The FDA is a little bit more stringent than the EMA. In 2017, in 2015, the EMA published in one of their papers that they included in the guideline that it's possible also to use the a reference drug outside, which is at the end like an American one out of an ICH area, and this is used in a clinical trial, and it does not have to be repeated using the reference drug out of the EU. But for the US, I strongly recommend, if you go first to the FDA and discuss it with them, if using the EU material is also accepted by them. We heard quite some discussion about having there really bridging studies between both originators.
Okay, thank you, Petra. Here's our next question. If analytical similarity is proven, why is a proper bioanalytical similarity still warranted?
It's again a very good question, because if two compounds are already analytical similar, why do we have a different challenge? Because it all depends on the capability of the binding. So it's the analytical in the analytical similarity testing, or in the Cmc testing, we test a lot of parameters, including potency as assays. In the bioanalytical similarity, we have additionally to the drug and the originator itself, we have also the biological metrics, which also can have an impact on that. And I think both work out together. So the bioanalytical similarity can never replace analytical similarity or CMC similarity, but it's an add-on. But also, even if it's not bioanalytical similar and it's analytical similar, it still does not prove that it will be biological similar. So it's at the end 3 steps in the corner. But as we've shown in previous slides, the bioanalytical part can give you maybe the additional hint. And often what we saw is that the characterization, what I showed on my when I presented the iceberg, the release testing is really the tip of the iceberg. And usually you include maybe there are 5 or 7 different type of assays. You test that this is the same, the new lot is comparable to the previous one. In the bioanalytical testing, we usually set up our assays using the same biosimilar lot and the originator lot, which is also used in the clinical trial. But this is not always the same which you use to demonstrate your biosimilarity in the CMC testing. And small changes which might not be picked up in the CMC testing or in the release testing of this lot, we might see in the bioanalytical testing. And I think we should use it more like a yellow card, like in soccer, or a warning flag. If bioanalytical similarity can't be proven easily and you see a challenge there, maybe take half a step back, go back, test, for example, also the biosimilar reference compound you used to demonstrate the analytical similarity to see if there's maybe something different between the two lots you involve in the complete discussion. But it's an add-on. Yeah.
Okay.
I could go on for ages with that.
Well, thank you very much for that. Here's our next question. Why do you think the generation of specific positive controls is of value considering cost and time?
This question I don't hear the first time. And it's often a starting point. And especially when for the originator, positive controls are commercially either polo or monoclonal are available. And yes, it's an easy point. We could use it, and if you can demonstrate the originator as well as the biosimilar both bind to the positive control of the commercial positive control the same, so this should be good enough. What we experience is really that when you use the positive controls against the biosimilar, which is really produced against the biosimilar, you ensure that all epitopes of the biosimilar drug are picked up, which you can't be certain when you use only the commercial available one for the originator. And additionally, what we learned that we achieved with the biosimilar positive control a 3 to 10-fold better sensitivity. And we also ensure that we include in our ADA testing all moiety of the biosimilar. And when we optimize the assay using this produced biosimilar, you can be certain for the submission that all antibodies generated against the biosimilar are definitely picked up. So this is really the most conservative approach and will already respond to several questions in advance. Because if you have an interference and you have the proper positive control, you can use this as a first argument saying originator as well as biosimilar act the same, therefore I have this part, especially when you do it in the one assay format. But to be really on the safe side, we also would recommend to produce in parallel with the biosimilar also the originator as a positive control in the same animal facility, in the same type of production, because then you have really a parallelism of both assays, and the assay is the positive controls really bind equivalent to them, and you also can pick up all the antibodies associated to the originator. Because there's always a risk of missing some positive antibodies. Yeah, so for us it was of benefit not to be having an easy life, but having already from an early time point as much information as possible in the bioanalytical assay, and it's really worth to spend the 3-4 months from the beginning.
Okay, thank you very much, Petra. Here is our next question. I think we have room for 2 more. So here's our next one. Here we go. Which top 3 topics should be considered for a bioanalytical PK method to support successful biosimilar programs? And what do you do when you can't prove biosimilarity during a PK assay setup? A little bit of a long one there.
Yeah, hope I get everything. So what are my first top 3 preferred when I set up an assay? I think first is really we try with the biosimilarity testing if we can achieve the single assay. And also for this, we have to consider the right assay format. So we need to understand the characterization, we need to understand really the biology of the drug and to have the proper setup there. And so what's the right assay format? What else? The critical reagents. consider that you have critical reagents which are available for the setup, but also for the complete development time. It doesn't help you when you have now the ideal lot of critical reagent and then it expires when you want to start with your second clinical trial. Or when during a long-lasting PK/PD trial, you have to restart and then it doesn't work out anymore. And also use the proper calibrators, said before, properly the biosimilar, and ensure with the CMC similarity that the head-to-head comparison works. And what to do when you can't prove biosimilarity during a PSA setup? Take your red flag, be warned, start discussing with your CMC folks. If there's anything you miss, go back to your assay, assess different type of maybe collection and detection reagents, different type of combinations, if you can improve the assay from that end. And because most of the time really something is underlying. And I think the discussion and open communication at this time is a key.
Okay, that sounds good to
Yeah.
me. All right, here is our last question, and it's a little lengthy here. In a previous paper, the two-assay approach is recommended for ADA assays. You recommend now the single-assay approach. What is the feedback from the regulators related to this?
Good point. I think in the first biosimilar programs we supported, we also applied the two-assay approach, and I think even if it was now published, I think it was online published December and 2019, but then January 2020 was unavailable in the community. This AAPS paper related to the ADA from the working group there, and there also we went back to, or there it was went back to the single assay. The current regulatory guidelines, the WHO, the EMA, the FDA, do not really give a clear hint how you have to set up the assay, and they just you have to have comparable assays. The majority now of the current paper, when you go back to the literature in the last 6 years, they all recommended the single assay, including this AAPS one. I think there were only very few one at the beginning who really went back to the two assay, because they have the same like in the PK assay. When you have two assays, you have to compare them. You have different assay properties having an impact on the results. And so the regulators who looked until today in the assays we supported, accepted it, and didn't have there any concerns with a single assay.
Okay. All right. Well, thank you very much, Petra. Well, I'd like to thank you. Thank you very much for those questions, audience members. We have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, the team at Celerion may follow up with you after this presentation. And if you have any further questions, please direct them to the email address that's on your screen. Okay, thank you very much, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. And get even more out of today's presentation by downloading the supporting materials available under the Handouts tab. And we have two there for you. One is a brochure, Leaders in Biosimilars, and then there's a white paper, Bioanalytical Challenges of Biosimilars. So thank you very much. I just want to say thank you again to Petra for joining us here for this presentation. Thank you so much for that insightful presentation. Okay, now we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you very much, Petra.
Thank you. Thanks for your time today. Thank you. Bye.
Bye-bye.
Bye.